I will get started with a few slides just to give you an overview of what CODEx is, how we use it, and then I'll go into the demo itself, where I'll actually show the tool and how it's best to use it if you're, for example, a pharmacometrician, or it basically depends on your needs and interests. So the purpose of the webinar is, first of all, to help you gain some familiarity with CODEx, so you can use it as a support tool for your ongoing projects. Second, I'd like to make sure that you understand when and how you could benefit most from using a CODEx database and using the interface. There are different parts to CODEx, and I'll get to that as we go. Then I'd like to give you some insight in next steps and how you can help us optimize CODEx further. So there are ways for you to provide feedback to us. The way we have developed CODEx and the way that we maintain it makes us very agile. So typically we can turn around requests very, very quickly. And it's important that you understand that that's super helpful and that it will also help you. And finally, obviously, since CODEx is a tool that helps you explore data from published data from clinical trials, or I should say reported data from clinical trials, it really connects to doing meta-analyses and model-based meta-analysis. So I'd like to cover a little bit of how those connect. But you'll see that as I show the tool, you'll immediately see how closely the two are connected. So first of all, what is CODEx? Well, CODEx stands for Clinical Outcomes Database Explorer. And it's, I should say it's a platform because it has quite a few components. Well, luckily for you as a user, you will only see one component, which is the online portal site. And you can find that site at codex.certara.com. And that's where we'll go to in a little bit when I do the demo. The platform provides you with a means to store, access, and explore summary level data that we have in our clinical outcomes databases. And these outcomes databases can be really, really big. Again, in the demo, I'll show you what they look like. But for example, a typical database has around 360 or 370 columns and anywhere between 10,000 and even more than 100,000 rows. So if you get that into your hand, let's say you export it as a flat file and you have to get started with that, it's pretty overwhelming. So what we have tried to come up with is a platform that allows you to very easily, with a click and go interface, very easily explore that. The content and the access is secured via an authentication and authorization system that is integrated. Again, that's not something that you will typically deal with, that you'll see. But initially, when we sign you up, let's say, I'll get to how that works. But when you sign up, you'll get an automated email from this system, and the system makes sure that you truly are who you are, and that you also can only see what you're supposed to see and not anything else. Right now in the portal, we have more than 40 clinical outcomes databases, and we're constantly expanding that. What can CODEx do for you in particular as an analyst? Because I think most of the people on the call are analysts. The basic idea is that you can become aware of the competitive landscape in the therapeutic area of interest, and actually beyond even your therapeutic area of interest. And that means that it allows you to really know your competition, know what they're focusing on. For example, what kind of endpoints, what designs are being used in a certain therapeutic area, what are typical results, what are the results for a specific treatment class or drug class, etc. You can even dive in a little more. So it's not just a general overview of stuff. You can actually do some exploration. So for example, you could have a look at correlations between endpoints, correlations between certain variables, covariates, and the interesting thing is that if you have access to multiple databases, then you could do that across therapeutic areas because, for example, some drug classes are studied in more than one therapeutic area. And even though in your therapeutic area of interest, the drug may only be in phase 1, it may have already been approved in another therapeutic area. So you could potentially look at side effects there. The efficacy endpoints may be and are likely different, but certain endpoints may be the same. Also, as you, all our consultants, for example, inside Certara, have access to CODEx, to all CODEx databases. That means that as they go through their own project, which may be a PK/PD project, which is not a meta-analysis, they may still access CODEx and put whatever they are doing in their project into a certain perspective of the clinical landscape and connect what they're doing to what other people are doing in a little bit broader perspective. Finally, if you are going to do or are doing a model-based meta-analysis, this is a really nice additional tool to have to kind of back up what you're doing and especially prepare for a meta-analysis. And obviously, before you get started with a meta-analysis, it's super smart to make sure it's not already done. So this allows you to really see what kind of research has been done, and if you are going to do a meta-analysis, what endpoints to focus on, what to expect. There we go. I mentioned meta-analysis. I typically assume that everybody knows what a meta-analysis or model-based meta-analysis is, but I thought I'd just add this slide for clarity. MBMA stands for model-based meta-analysis, and meta-analysis in general is a quantitative analysis of reported research results, and those results can be at an aggregate level or at a individual data level. And so you might be looking at summaries of patients or individual level data from patients, and there's a combination is possible as well. If you look in PubMed, or if you go on the web and you look for meta-analysis, meta-analysis reports, which are typically called systematic reviews, and then if there's a quantitative part to it, that part, to be strict, that part is the meta-analysis. Most of them are meta-analyses of summary level data, and the reason is that a lot of people don't share their individual level data. Also, most published meta-analysis used to be pairwise meta-analyses. Pairwise meta-analyses are, and I'll show you that in CODEx itself as well, are meta-analyses that look at things in pairs. So you could look at a certain treatment versus placebo, or a certain treatment versus another treatment, but you look at them one at a time in pairs. And that's called a classical meta-analysis or a pairwise meta-analysis. Nowadays, we see more and more network meta-analyses, which there's a lot of similarity between the methods, but you're basically looking not in pairs, but you analyze an entire network simultaneously. That has a number of advantages. And model-based meta-analysis is essentially a network meta-analysis that's slightly more complex in the sense that it can integrate, if it's feasible, dose response models or pharmacological models. It can be Emacs models, it can be anything. But whether you can do that or not depends obviously on the data that you have at hand. And MBMAs can be used throughout a drug development cycle for multiple purposes. You can use it for decision making for a portfolio, for trial design, to choose which endpoints you may use when you go to market, etc. So we're almost at the demo. Let me take a quick sip. So how does CODEx work? Well, as I said before, the main moving part for you as a user is the portal or the CODEx website. And that, first of all, allows you to explore, what we have out there. Your company can purchase a license for access to one or more databases. And when they do, the access to one database comes with a number of named users. So right now it's 3. Additional users are an option, and many companies do have additional users. Before your first use, you will receive an email from what's called Okta, and this is this authentication and authorization system. And that way, you'll be recognized, and once you log in, you, as a little bit of background information, CODEx was developed with R and a tool called Shiny, and in order to make sure that that gets stored securely and you can access that, we use something called an RStudio Connect server. Once again, you don't see that because everything is run via the website. But the moment you log in via Okta to the website, you also log in to the RStudio Connect access, and so that gets authenticated as well. So we have kind of 2 levels of security in terms of making sure you access what you're supposed to access, and that is at the website level as well as at the server level for the R content. When you log in, you will see something called all CODEx databases as well as your CODEx library. And your CODEx library will be at the top, and it looks a little bit like what you see here at the bottom right. And depending on the licenses that you have for the databases, you'll see 1, 2, 3, or maybe even 30 databases. It depends. But however, if you don't have access to all databases, you can still see which databases are available in the part that says all CODEx databases. So that's in a nutshell how it works. So let me stop sharing this part, and then we'll check it out in the tool itself. Let's see, share my browser. All right, let me move this out of the way a little bit. There we go. So when you go to codex.certara.com, this is the website that you'll be landing on, and it's very, very simple. I'll take you through it very briefly. Once you log in, and just to make everything a little bit quicker, I have logged in, but there's a little button on the top right, and when you click on it, you will there'll be a button that says please log in. In my case, I've already logged in, so it asks me if I want to log out, which I obviously don't. You'll see 4 buttons or 4 menu items at the top. Because I logged in, I also have my account here. On the home page, you'll get a brief overview of what it is that we're doing, what CODEx is, who's doing all this at Certara. There's an additional page about who's doing it, not only who designed it, but also everybody who's working on it, and so you can briefly see who's guilty. And then when you click on CODEx, which is probably the most important part, you'll see, in this case, at the top, you'll see your CODEx library. And because I have access to everything, and all the Certara consultants have that as well, I only have my CODEx library. So I don't have an overview of all the databases because everything is already in here. But most likely, in your case, if you would have access, you would see a number of databases here, and then there's another heading that says everything. You can click on Contact Us. That will give you a form that allows you to connect with us, but you can also connect with us inside CODEx itself. So from here, you can click on any gallery item. You can also search for specific databases. So I can search for lupus, for example. Or let me do something that's in my view right now. I can go Chrome, Crohn's, and that will get you to stuff. And then when you reset it, it'll get back to all the databases. When you click on a database, you'll get to a summary page. From there, you can return to wherever you were. You can look at a summary PDF. So let me open that in a new tab for a moment. There we go. The summary PDF will give you a very brief overview of what's in the database. So you can always do that. You can always download a PDF. It gives you an idea of how it's made, and it gives you a contact, who to contact if you're interested in it. But everything, actually, there's even a little more detail on the website. So when you click on one of the pictures at the bottom, the graphics, you'll see the first page of CODEx itself. And then there are a few additional items, and how many depends on the database. Any item that you click on will open up in a slightly bigger window that you can close here at the top right. So most importantly, obviously, is accessing the database to make sure that that is a little bit quicker. Oh, it's still open. I already did that. This is a nice database to share with you. I also opened up the CODEx COVID database to give you an idea of what that looks like. But let me go here. So because I already opened it, and this is a nice feature, I think, if I say so myself, if you've opened something already, and let's say I've been in this part of the tool and I close it, it will remember. If I open it again, it will remember where I was. So that's kind of nice. The reason why we did it that way is because depending on how big the database is that CODEx is loading, it may take anywhere between 10, or between 5 and maybe 25 seconds before everything is really loaded. And we're trying to speed that up, but yeah, some of these databases are really, really big. So let me give you an overview of how the tool is set up, how it's formatted, and how you can use it optimally. First of all, there's a top bar. So when you, and everything opens up in, it's called an iframe inside the website. So when I click this, there's a little overlay. This white part is an overlay. When I go out of it, I come back to the website. There's a top bar inside this overlay that has a number of tabs. And what we've tried to do is keep it simple. If you go from left to right, you can't go wrong. It goes from simple or general to more specific stuff. Also in the top bar, there's a little button that allows you to provide us with feedback. And you can see at the bottom left of my screen, hopefully, that the email will go to CODEx feedback or codex-feedback@certara.com. So if you come across something as you explore the data that looks iffy or something that you would like to see differently, then just pop us an email. We'll typically get back to you within 24 hours saying thank you for sharing this with us. And because this is made in Shiny, it's very easy to adjust things, to add another button or to add something to a drop down. And we also use something called continuous integration and automated deployment. So the moment that we adjust things, if we want to, we can have all these databases updated with one click. So all the code that we use is generalized. And that means that the code that we use to create the Codex interface for NASH, the one that we're looking at right now, is the same as for any other database. There's one exception, and I'll get to that. So we have this top bar at the top, and then we have a sidebar on the left, and a main body. And when you open up the interface, the main body has 5 general parts: an overview, a summary of what you're looking at. And the summary says complete and filtered. Filtered refers to the filters that we have in the sidebar. And I'll get to that in a little bit. Then we have an overview of where these trials were, or the studies that were done. Mostly will be trials, where they were done. Now, and this brings me to a cool feature that we have throughout Codex, and that is that we have reactivity from our filters on the left. So whenever I change a filter, and I'll show you in a little bit, whenever I change a filter, anything in the body, or anything really except the database part, but anything else in the interface reacts immediately. So we have reactivity to filters, but we also have interactivity of our tables and plots. And that means that you can interact with that specific item, and it will adjust to whatever it is that you're doing with it. So most of these tables and plots, you can click on and you can hover over. And so, for example, when I hover over this simple, like a heat map, pretty much geographical, it shows me the information. It shows me how many study sites there were in various places in the world. But you can also zoom in and zoom out, so you can interact with it. And the same goes for, let's see what we have over here. So I can see the most common efficacy endpoints here at the top right. And when I hover over it, I can see in how many studies it was reported. And obviously here on the front page, everything is very simple, but it gets a little more complex as we go through the interface. And we'll do that in a minute. But probably the most important, the most important thing for you are these filters here on the left. We already saw that here it already says filtered, and to make the loading a little bit faster and to make things a little easier for you, we have some data settings that we filter right off the bat. And take a quick sip. So when Codex is loaded, or when the data are loaded in Codex, I should say, in principle, we select primary time points only. So if you have studies that report longitudinal data, we deselect them for most of the plots, except for the time course plot. You'll see why, because then if we wouldn't do that, you wouldn't see a time course. And the same goes for a primary statistical analysis. Is it an ITT? Is it per protocol? And also primary strata. So often there are multiple strata reported. We pick the primary ones only when we start. When you can unclick these and automatically the interface will react. But most of the time, you want to leave this just as it is. Active filters shows you which filters are active. So for example, the way this all works is, let's say I have a certain filter variable. Let's say I'm not interested in non-alcoholic fatty liver disease. I unclick this and I keep NASH selected and I say apply. And now I see here that there's an active filter, and I also see that the number of studies which were originally set at 156, and I can, if I reset it, see it automatically react, it comes back to 156. It changes. For some of the variables that you have here, let's see, for example, randomized drug, if I'm, or let's do drug class here. If I'm interested in a specific set of drugs that I can select here, you may want to filter on trial level. So if I only select these, I will exclude all the rows that have drug classes, that have treatments in a drug class that is not in here. Those will be deselected. But I may want to include the entire trial because I want to look at comparisons. In that case, I could click filter on trial level and apply. And that way everything gets filtered on trial level. Right now, I have 7 studies left, which is very little. So let me reset it. Once you reset it, it comes back. Oh, hold on. Reset all filters. It comes back to where you started. So, but let's narrow it down a little bit. So let me go to NASH only. I'll apply that. And maybe we could say, hey, we're only interested in maybe in parallel trials, not single arm trials. I apply that. Let's see. About 74 studies. So that's how the filtering works. The next tab here at the top is the database tab. This is the only part of CODEx that does not react to the filters, because we'd like you to understand what is in the database itself. There's a general summary. You can download the PDF of this. Every table has these buttons that allow you to download a PDF, a CSV file, an Excel file, and copy stuff to the clipboard. There are some additional tabs here, or sub-tabs, that show you what the included references were. In this case, we have 195. How many references were searched, which is obviously a lot more. These tables you can also interact with. So if you're looking for, let's say, a specific author, you can, Mr. Harrison, for example, you can look that up here and it will interact. You can select stuff. Or maybe you are looking at, at a specific publication year, you can select based on that. And also, if you found a study, let's say in search references, that may have been excluded. So right now I just selected everything that was excluded, and I want to know, well, why was this study excluded? I can click on this link and it will take me, if there's an entry in PubMed, it will take me to the reference as it is in PubMed. If you're lucky, you can look at the full paper. We do not provide you with the full papers for copyright reasons, obviously. One other thing that's important to understand is that when we build these databases, we build them in 2 steps. First, we create what we call a source database. And the source database is a database with all the references. So we build that first. Based on that, we can already make a first selection. We can already see from the abstract that the design was completely different, for example. And from there, we build an outcomes database, and you can download them from CODEx. You can download both of them, and both have specifications. So there's specifications for the outcomes database, and there's specifications for the source database. So that's what you'll find in this database tab. Now, when you go beyond these two tabs, it becomes more of an analytical thing. So first of all, you may be interested in what kind of studies were done. You can select specific studies that you may be interested in, and this gives you a very brief overview, typically including the abstract if that was reported. And you can look at the individual study. We also have study summary tables that has a single row per study, and we provide you with some information in there already. Move this out of the way for my screen. Let's see. There we go. But again, it's interactive. So if I don't want the study phase in there, I can take it out. I want age and males, but I do want whether a study was blinded in there, for example, and how it was blinded. So you can add that. You can also sort it. So if I want to sort based on the number of patients that were included in the trial, you can very easily do that. There are covariate plots that we have here. Some of them are very busy. So here we have, in how many studies was a certain drug used. You can select number of studies or the number of subjects in an arm. Because some of these are very busy, we've taken out the labels, but because we can make it interactive, you can hover over them and you can see which drug it was. You can also see the number of patients, but if you select, you can select many different things here. But for example, when you select something like age, which is continuous, we conveniently put that in subcategories for you, and so you can see by subcategory how many, typically in the trials, in which subcategory do you find most of the patients. So that's what you can do in these study covariate plots. We also have protocols, for how we develop databases. So you can see we typically follow a process as it is outlined in what's called a Prisma statement. So you can go over the protocol here, and we augment all our databases. And I'll explain that briefly because it's actually very important. Obviously, when you find studies, whether it is, we look in reference databases like Medline, or you will mostly probably use PubMed for that. But PubMed, there are other reference repositories out there that we use, but we also use regulatory websites. So it could be clinicaltrials.gov, it could be the FDA website, the EMA website, and we even look at abstracts from conferences. Once you have these references, it's not so hard to translate that qualitative information into something quantitative and put it in a database. But the way things have been reported across these different studies is typically very different. There may be similar endpoints may have different units. For example, covariates may have different units. Obviously, the doses are different. Dose units may be different. And if you do not take that into account when you create plots, your plots are going to be ridiculous. And I've seen plenty of ridiculous plots in presentations coming along. And that's simply because people haven't been exploring stuff in detail, haven't taken care of standardizing certain things. And so in our augmentation process, which is mostly automated, so we use code to standardize units. We use code to create additional columns and to calculate. So for example, if you have a baseline reported and you have an absolute value reported, you can calculate change from baseline. You can also calculate % change from baseline. So all these calculations are things that we take care of already. And everything that you see inside CODEx is based on an augmented version of an original database. So everything it's ready for analysis, with one exception, and that is that you may have to select specific, before you do like a formal model-based meta-analysis or meta-analysis, you probably want to select specific strata or general strata. But you want to make sure that a patient is not represented more than once in a group that you're analyzing. So talking about exploring and analyzing, let's have a quick look at some of the analysis tools that we have inside CODEx. So first of all, when you click on exploration, you'll see a time course plot. And in this case, I have only crossover and parallel studies selected. So you will always see multiple arms in here. I'd like to point out one more setting here at the bottom left, and it says default measure. We have 3 measures in our databases. One is called value, one is called change, and the other is called % change. A value is the absolute value at a specific time point. Change is the change from baseline, and % change is the % change from baseline. By default, it is set to value, which means that when I look at baseline here at time 0, there may be, the baseline is not always the same in every group. So if you would choose here change, you will automatically see everything change on your screen, obviously, right? Because everything is reactive to these filters. And now in my time course plot, everything starts at the same point, because what we're looking at is a change from baseline, and by very definition, the change from baseline at baseline is 0. Right now, the way we have pages in these time course plots, because we have to, these plots are interactive as well, right? I can hover over it, it gives me information, and that is why our pages work like this at the moment. So they work with a slider. That way you can move through them. You can choose different endpoints here. So right now we're looking at ALT. If I click on AST, it'll automatically change right in front of you. Next up are, this is actually nice. It's not supposed to happen, but it's good that it happens. So I can show you, this sometimes happens and is due to there's a small bug in the plotting code that we use, the plotting package that we use. But over here, it says on the left, it will say use Control plus or minus to adjust the interface formatting to your screen. So if I press Control minus and maybe one more time plus to get to the original, you'll see what you're supposed to see. We know about this bug. It's why it's actually nice that it occurred right now, so you don't have to provide us feedback on it. We're working on it with the authors of the package. You can look at some correlations. First of all, between covariates. So here we're looking at age and ALT. Maybe you want to look at AST and age, and it should be pretty similar. You can adjust the smooth. If I do 1, it's a straight line. And you can make that a little more fancy. In this case, that wouldn't be necessary. You can look at correlation between endpoints themselves. So right now, I'm looking at ALT and AST. They're highly correlated, obviously. And one other thing that's nice is you can look at it descriptively and comparatively. So right now, I'm looking at, as we saw here, the change from baseline. But I may want to look at it in terms of a change, the change from baseline from control. In this case, that is for ALT or AST, but that will give you a slightly different picture. And it's important to understand the difference. That difference, I'll show you, becomes clearer when you look at forest plots. In the last part of the exploration is actually a very important table, and you can create different types of tables here. But I may, for example, be interested in, okay, what are the most important endpoints that are being studied, or which ones are studied most often? So I can click on studies here, and it automatically sorts. So I can see which are the most common endpoints. I could look at it in terms of patients, which is typically very similar. I can look at specific publication years, although this is numerical in this case, not numerical, excuse me, or maybe specific endpoint categories. So maybe I'm interested in, let's see, biomarkers only, or maybe I'm interested, it could be that you're interested in adverse events only, for example. I can click this away. So this is interactive as well. And then the last part is the meta-analysis part, which we call it meta-analysis at the moment, but there's no, at this stage, but it will be there very soon. There's no automated meta-analysis in there in the sense of giving you summary level weighted means, for example. This is still, it's CODEx, it's an exploratory tool, so it gives you exploratory information. So here is a network plot, and you, because we don't show labels initially, because that can be really busy if you have a lot of trials, but in this case it looks good. You can see which, in this case, which drugs are compared with which other drugs and how they are connected, and some are not connected. So this network that we see here on the left is a connected network. This is disconnected from the main network, and these are all disconnected, which means that if I do a model-based meta-analysis, I could never compare any of these interventions with these. Again, this is an interactive plot, so if I want to, I can zoom in, right, on specific parts of this. Here at the top right, you can, if you click on reset axis here, the home button, you get back to where you were originally. But also when you hover over it, it gives you additional information as well. We have a forest plot that we're going to replace soon, actually, with something that looks a little bit nicer, and that's also going to be used in our automated meta-analysis tool. But over here, you can see change from baseline for ALT. You can see the treatments and controls. It's sorted by study. And here it says descriptive. And I promise you to come back to that. If I click comparative, I'm looking at now a mean difference. So the mean difference between the intervention group or the index group and the reference group or the control group. If it would be, so here it says for continuous comparisons, which ALT is a continuous endpoint. I'm looking at mean difference. For binary, you have some options. If it would be, say, death, then you could choose odds ratio, risk ratio, or a risk difference in this case. And again, this will react. If I choose different endpoints, it'll give me, the graph will automatically react. You can hover over it. Once again, I can select specific part of it to zoom in. You can also add, let me show you, because that's sometimes goes off the screen. You can add and take away specific columns here in the plot itself, here on the left-hand side. Finally, there's a funnel plot, which again, you can zoom in, you can hover, which gives you some information on, first of all, some heterogeneity. Are some studies falling outside of my, these are pseudo confidence intervals. And also, typically this plot is used to get an idea of positive potential selectivity in the publication of studies. So ideally, this plot should be fairly symmetric around this line here. That's really it. Finally, there are some settings which, if this wasn't super clear, there is a manual for you that has a lot of the information that I just explained in there. So you can use that. And there are some settings here. There's some version info, some settings for colors. If you're colorblind, let me see if I can show you that. If I go to my time course plot, you can see the type of colors that are shown here. If I click colorblind friendly, and I go back, it just changed. So it'll change to a colorblind friendly palette. Put that back. There's some stuff for font sizes, some formatting things that you can adjust. For example, for the time course plot, maybe you only want 2 panels, and it'll change. But we've kind of hidden this in the back because you wouldn't use this, probably wouldn't use this very much. But what you will use are these download buttons. So if there are multiple versions of a database, they will be available for download. What you see in the interface itself is always the most recent version. So if you do want to explore an older version, you can download it and explore it yourself. But in the interface, we will only show the most recent one. You can download the databases, which will be Excel files, or you can download analysis sets. And analysis sets, the database is the full database. An analysis set is, is dependent on whatever filters you have active here on the left-hand side. So if I download the analysis set here, it will download a CSV file of Nash, and it's pretty big, as you can see. And once I'll pause this, there we go. But it will, how big it is will depend on how restrictive your filters were here on the left-hand side. And if you download it, you can also download an overview of the filters so that you can store that and know, okay, for this data set, these specific filters were active. Last quick thing, so we also have a COVID database that's been used a lot, and I do want to show you that because this is the only database whose interface is slightly different. And when you go here into meta-analysis, the options that you have are a little bit more extensive. And we use this database to experiment with a few things. So you'll see some stuff in there that may become available in other parts. So there's pairwise meta-analysis. there's network meta-analysis in there as well. The network plot is slightly different. And you can also look at specific risk factors. For example, in this case, it's not available, but you can look at specific risk factors for an endpoint, and you can do that again descriptively and comparatively. So these two items here at the top are slightly different than what you see in most, actually in all the rest of our databases. Again, which you can find if you go to CODEx and then look at all databases or your CODEx library, and you'll get a good overview of what's available in there. Let's see. That is all I had for you this morning. Let me stop sharing my screen. Now I think, Suzanne, you may have gathered some chats already, or maybe not, but if you have any questions, you can type them in the chat and I will try to answer them for you.
Okay, thanks, Leon. Hi, this is Suzanne Minton. Good morning, everybody. Leon, it looks like we have our first question. Someone says that they're an infectious disease physician, and this person wants to know if there's work being done to develop more infectious disease antimicrobial databases.
Yes, but I have to admit that most of the work I've done in terms of meta-analysis has been in immunology, in Crohn's disease, ulcerative colitis, and metabolic diseases, so diabetes, for example. But the way we try to develop our databases is through interest from our clients. So the way these databases, and this is actually nice that this question was asked, the way these databases have come to be is often through a request from a client. So if there is interest in a specific therapeutic area, somebody may ask a question like the one you just asked, and we'll look at, well, if this is an area of interest that is meaningful to a large group of patients, and it's commercially meaningful to people in the industry, then it's useful to develop this database. And what we often do is that we say, hey, we understand that you are interested in this, but we will take on financially the development of this database, and that way we keep the intellectual property, and you can purchase this database for much less than the cost that the amount of investments that we had to make to develop this database. I have to admit, at this very moment, I do not know what is cooking in the kitchen, so to speak. But if you, after this meeting, if you connect with me, let me get rid of this. If you connect with me, I'll make sure you get in contact with the person who's most familiar with which databases are currently not shown here. You can obviously use this to see what is developed, what is available, but I do not know what is cooking in the kitchen, so to speak. So I can put you in contact with somebody who is really familiar with that. I don't think Nancy's on the call at the moment, but we'll get you in touch with someone.
Great. Leon, someone wants to know whether CODEx can be used to search for adverse events reported when taking a particular drug.
Yes. Here we go. Where were we? We were looking at NASH. So let me go here. If I understand the question correctly. So let me see. So now because I closed the entire page, it's loading again. This is good for you to see as well, even though I'll while this is loading, I'll try to answer the question. So yes, we have efficacy endpoints as well as safety endpoints. If you're interested in a specific safety endpoint, you would, let me wait till its interface completely loaded. There we go. You would go to exploration, you would look at overview tables, and you would select endpoint, and then here endpoint category, I look at adverse event. If I'm looking for example, cramps, I can type cramps in here and I can see muscle cramps, abdominal cramps. So
Leon, I don't think you're sharing if you mean to be sharing.
oh, good point. All right, hold on, let me share. Thanks, Suzanne. Here. I'm assuming you're seeing it right now. So let me take this away again. So you would go in CODEx, you go to exploration, overview tables, and I'd select endpoint. And here, if I want to, I can already go adverse event. I click on it. Now I get all the adverse event endpoints here. They're sorted alphabetically. I can type something in here like cramp, and I can see, okay, I've got muscle cramps and abdominal cramps. The way we build our databases, we try to integrate every endpoint. We may change that in the future because the database gets so bloody big that, and you know, obviously not everyone is interested in all these endpoints. Let's see. Okay. Let me stop the sharing. And here we go. So yes, every endpoint that is reported gets integrated in our database. So certainly all the reported adverse events will be there.
Great. Someone wants to know if they're writing a publication, how do you reference CODEx in a publication?
Wow. I haven't thought of that yet.
Yeah, couldn't you just put CODEx Certara, Princeton, New Jersey? I mean, I would think that would be standard.
Yes, but I will check on that. And again, Suzanne, if you can take a note of who asked, I can get back to. Yeah, but that's a good question. If you use it, it actually has been cited already in 2 publications, and people did like what you said, Suzanne. So they gave the website and referenced Certara, where Certara was. But it's probably a good idea to clarify that. Thank you for that question.
Someone wants to know if we have, if CODEx has databases on pain indications, for example, osteoarthritis, migraine, etc.
Yeah. So again, let me share my Chrome. No, stop this one. Yes, we do both. So we have neuropathic pain and osteoarthritis pain. If you go to the website, you can already see this. If I type in pain, these are the 2 databases that we have. Neuropathic pain is post-herpetic, I think, is one of the indications.
Yeah, post-herpetic neuralgia, fibromyalgia.
Exactly.
And diabetic neuropathy, I think.
Yeah, yeah. And then we also have osteoarthritis pain. If you click on any of these, you can already see, or there's actually, let me get rid of this. There's a summary here that shows, how many studies, what are the most common endpoints. So that information, you don't have to log in to see this, just to clarify that. I hope, yeah, it doesn't matter. There we go. I log out here. Yeah, I really do. Oh, that's interesting. Let's go here. But yeah, even when you're not logged in, you can have access to the basic information. So yes, we do have several databases related to pain.
Great. Lan, this looks to be the last question from the audience. Someone wants to know whether it's possible to filter the databases by targets.
What is
I think they mean like for pharmacological targets. So can you filter by like a certain GPCR is what I'm asking if, please clarify if the person, if I'm not getting that right, but I think that's what they mean.
Yes, yes, you can. So when you, let me see, am I still, yeah, I'm logged out, but let me log back in. Let me do this for a moment. There we go. Nope. When you go into CODEx, well, yeah, let me not do this via the screen. Let me just explain this. So you can filter on everything that is a column, almost everything that is a column in the database and is a covariate or is an endpoint. So the filters on the left, if the target, and typically they are, if that is included as a covariate in the database, then yes, you can filter on it. And if there are, for example, some targets may be in categories, then the filter itself will be categorical. So it'll be checkboxes. If it's a continuous thing, then there'll be a slider. So the answer is yes, you can filter on that. And depends a little bit. You have to be careful, right? Because some of these may be in strata, and then you want to filter on it, but you want to make sure that you do it at a trial level, so that if the targets that you're talking about is if there are multiple, if it's categorical in a trial, then you may want to capture all of them. But yes, it's possible.
It looks to be all the questions from our audience. Thank you so much, everybody.
Yep. Well, great. Once again, thanks for dialing in. And if you have any additional questions, you can contact us either via the website, or if you're exploring CODEx, you can use the button on the top right. But thanks for listening and dialing in, and have a good rest of your day.